Hello everyone, this is Sukanta Nath from India. I have completed my PhD in biotechnology and working as a research scientist at Atal Bihari Vajpayee Regional Cancer Center. It's a state cancer hospital. It's in Agartala of Tripura. So at present I am working on HPV. Today I am going to present a small topic on HPV. Its prevalence and age-specific distribution of genital high-risk human papillomavirus among women in Northeast India. So cervical cancer imposes a huge global burden because of its high mortality rate. The main pathogenetic event that contributes to the development of cervical cancer is a human papillomavirus infection. Cervical cancer is the fourth most frequent cancer in women worldwide after lung, colorectal and breast cancer. However, it remains a major health concern, especially in specific regions like India. Cervical cancer ranks as the second most prevalent cancer among women in India, accounting for approximately 18.3% of all cancer cases, and is linked to several factors including HPV infection, age at marriage, number of pregnancies, genital hygiene, nutritional status, usage of oral contraceptives, smoking, etc. Now, HPV is a DNA tumor virus that induces epithelial proliferation at cutaneous and mucosal sites, and in contrast to many other malignancies, women are usually affected early during their reproductive period. HPV is implicated in the development of cervical cancer along with several other cancer types including oropharyngeal, vulvar, vaginal, penile and anal cancers. Presently, it is established that there are over 200 unique strains of HPV with roughly 30 to 40 strains affecting the human genital area. Among these, 15 strains have been identified as being linked to cervical cancer. Because of their association with cervical cancer and its precursor lesions, genital HPV types can be classified as high risk, that is oncogenic, or low risk, that is non-oncogenic. Globally, high-risk HPV type infections are related to more than 90% of cervical cancer occurrences. Persistent infection with HPV is an essential contributor to the development of cervical cancer and the formation of precancerous lesions. Moreover, it has been observed that 10 to 15% of women over 35 years old with persistent HPV infections face an elevated risk of cervical cancer. However, the incidence of cervical cancer is also determined by the type of HPV, the characteristics of the host, and environmental factors as well as their mutual synergies. In northeastern states of India, there has been limited research on the molecular prevalence of HPV, mainly due to the scarcity of diagnostic resources, especially in Tripura. The present study was aimed at determining the prevalence and age-specific distribution of high-risk HPV in Tripura, that is in India, since testing for HPV is now an important tool in managing women with cervical precancer. To the best of our knowledge, this is the first molecular report from Northeast India with a US FDA-approved Cobas 4800 system that offers high-throughput, fully automated, real-time PCR with individual identification of HPV 16 and 18. And pooled detection of 12 other high-risk HPVs, that is 31, 33, 35, 39, 45, 51, 52, 56, 58, 59, 66 and 68. So this study aimed to determine the prevalence and age-specific distribution of high-risk HPV infection in the Northeast Indian female population. So this study was conducted in accordance with the Declaration of Helsinki and was approved by the Institutional Review Board of Atal Bihari Vajpayee Regional Cancer Center, Tripura, India. Informed consent was obtained from all participants before sample collection. So women over 18 years or at least 18 years old attending the gynecology department of Agartala Government Medical College and Govinda Vallabh Pant Hospital of Tripura who attended for a gynecological examination were approached to conduct the molecular HPV testing. A cervical swab specimen was collected from suspected patients. Samples were transported to the molecular laboratory of ABBRCC for HPV DNA analysis. The Cobas 4800 HPV test is a qualitative assay that provides specific genotyping information for HPV types, that is 16 and 18, while concurrently detecting 12 other high-risk HPV genotypes as a pooled result. This slide I am showing you just to let you know where exactly we have completed this study. In India, if you see, there is a red colored state, it is called Tripura. If you see in Tripura, it's in the northeastern part of India. In Tripura, if you see, there is one blue colored district, it's called West Tripura. So in that district, this Atal Bihari Vajpayee Regional Cancer Center is situated. It's in the capital of that state. So the Cobas 4800 system consists of two parts: a Cobas X480 sample preparation instrument, which simultaneously extracts HPV and its cellular DNA, purification and mastering preparation, and the Cobas Z480 real-time PCR analyzer, which amplify the target DNA by PCR and nucleic acid hybridization for the detection of 14 high-risk HPV types. The entire test procedure is monitored by an internal control beta globin. Although as a standard protocol, a set of HPV positive and negative controls were included for every sample run. The statistical software programs SPSS22 and APinfo 2000 were used for all statistical analysis. Statistical significance was considered to be p less than 0.05. So we tested total 570 samples for genotyping by Cobas 4800 real-time PCR. The overall prevalence of HPV detected in this population was 10.3%, or you can say in 59 total out of 570 samples, which was consistent with findings from other studies in Asia and Western countries. HPV-16 emerged as the predominant type within the population group, identified in 32 samples out of 59. In contrast, HPV-18 was detected in only 3 samples out of 59. The pooled HPV types were determined to be 31 out of 59 samples, positive samples. HPV-16 exhibited co-infection with other high-risk HPVs in 7 samples out of 32 samples, while co-infection was not observed in HPV-18 positive patients. So this slide I am showing you because we have done comparison also with other studies. So for example, in our study it was 10.3% prevalence. In Mexico, Pons et al. They have shown 14.5% prevalence. In Finland, Leninonen et al. They have shown 7.5% prevalence. In Spain, Chesselseg et al. They have shown 14.3% prevalence. In British Columbia, Moore et al. They have shown 16.8% prevalence. Although for few studies methods are different, but almost similar results we have found in few countries also. In our investigation, we observed an inverse relationship between age and HPV prevalence. Among women who had HPV infection, the age group distribution was 25.4%. That is for 18 to 30 years, 32.2% for 30 to 40 years, 28.8% for 40 to 50 years, 8.4% for the age group 50 to 60 years, and 5.1% for 60 to 70 years. The findings show that women in the 18 to 30, 30 to 40 and 40 to 50 age groups had a considerably greater prevalence of high-risk HPVs than women over 50, although this difference was not statistically significant. So HPV is common among women in Tripura, and the prevalence of HPV-16 is notably the highest, followed by other high-risk HPV genotypes. Conversely, the occurrence of HPV-18 is significantly lower. About age, the highest HPV prevalence was found in the 30 to 40-year-old group and the lowest was in the 60 to 70-year-old group. However, the findings of this study cannot be generalized to the whole country due to the well-known significant regional and population variations in HPV prevalence. The escalation of cervical cancer cases presents a formidable challenge for tribal and low-income communities in northeastern India. There is a considerable deficiency in information regarding the prevalence of HPV genotypes, which is vital for developing an effective plan to eradicate cervical cancer. The timely diagnosis of cervical cancer is often hindered by insufficient awareness and the absence of effective screening initiatives. Therefore, the implementation of robust HPV infection prevention programs can significantly contribute to the prevention of cervical cancer. So thank you so much everyone. So I know it is a small presentation. At present I have this much data only to share with you. I'm sure in future I'll be getting more data, or if I work with some other cancer markers, then I'll be able to share more data and it will be a big presentation. So anyway, take care. I hope you like my presentation. Thank you so much.